talk about pain management testing, why it's important, why we do drug testing for pain management patients, and then talk about some implementation issues relative to the approach for drug testing, a potential algorithm for how to implement drug testing, and then talk about interpretation of results. Because at the end of the day, that's what we need to do best, is interpret our results. Both positive and negative results are going to be expected, and determining whether those are appropriate in the clinical context is very, very important. At the end, I'll also make some comments about using urine drug testing for the purpose of drug adherence versus dose and dosing adherence testing. As you'll come to appreciate, the primary purpose of drug testing in pain management is to verify that patients have taken medications they should, but abstained from medications they shouldn't take. And many people ask, can we also determine if the patient took the exact dose that was prescribed to them at the prescribed frequency of dosing? And so we'll talk about that a little bit at the end. So why drug test in this population? And what we're talking about are primarily non-cancer chronic pain management patients. The drug testing is becoming standard of care as a tool that supplements self-reporting, behavioral, and clinical monitoring. So as I said, this really is to help improve patient care from the perspectives of ensuring adherence with prescribed therapy, but also to assure abstinence from unprescribed therapies, including other prescription drugs as well as illicit drugs. And all this works together to promote patient safety. We know that routine drug testing does help reduce the use of illicit substances and prevent drug diversion, that is, selling drugs on the street or hoarding drugs, things of that nature. So urine drug testing for pain management purposes is very important. Protects the patient, the payer, and the physician. So now you all know it's important to do. How do we do it? How do we select the best drug test? Well, you'll find that the pattern of drug testing mimics traditional drugs of abuse testing in that there's typically a drug screen that looks for lots of different drugs, and then also confirmatory testing to follow up on things that were positive in the screen that require further attention, or perhaps things that were missed by the screen. So all in all, the pattern is similar to drug abuse testing, but really the content, the specific things we do should not mirror traditional drug abuse testing because the objectives are so different. The testing needs for pain management patients are going to vary based on the population of patients that's being tested. That is, what drugs those patients are prescribed, what their risk profile looks like, the formulations of drugs that those patients are being prescribed, whether it's primarily transdermal patches or oral medications or other types of formulations. Urine drug testing is probably the cornerstone right now, so urine is the preferred, most common specimen, but at times other specimens, such as blood, can be very useful in a pain management population. And so knowing that alternate matrices are available for drug testing is important. Certainly the performance of the testing is very, very important, so knowing what the sensitivity and specificity is of a test, as well as whether or not quantitative results are generated, can be very important. In general, in pain management, we're trying to avoid false negatives as much as possible, so the cutoffs tend to be much lower than they would be in a traditional drugs of abuse testing profile. So in many ways, a pain management drug testing approach is going to be a hybrid of therapeutic drug monitoring principles as well as drug abuse testing principles. Now, in terms of screening, the most common platform is an immunoassay, and specificity is a major concern when we talk about immunoassays. So an immunoassay is based on an antibody, which is raised against a specific molecule. And so the antibody on which an immunoassay is based is critical to defining the specificity of that particular test. The cutoff concentration and the calibrator are also important. Now, if the specificity is perfect, so 100% specific, then we get good results and we can be very confident in our positive or negative result. However, we know that most immunoassays don't have perfect cross-reactivity with the drug of interest, so we may end up with false positives or false negatives. The next couple of slides show some examples of drugs that may not be detected by common immunoassays and therefore lead to false negative results. And then also some drugs that may be detected with good cross-reactivity and lead to false positive results. So false negatives and false positives are very important. The cards that you each should have received have a link to the website which includes all of these slides, so if you want to look at them in more detail or consult some of the references included here, that is available. In any case, the take-home message is that test performance characteristics will dictate your drug detection capabilities, and that is whether a result is positive or negative. Other things that are real important include the specimen itself, what kind of specimen it is, the quality of it, and when we're talking about urine, that may be the dilution or the pH, the timing of collection relative to the timing of drug use is also very important. So if a specimen is collected too long after drug use, or if that specimen is very dilute, we could very easily miss a drug that is present but at concentrations below the cutoff concentration. Okay. Drugs themselves and various formulations have different bioavailabilities, different delivery rates, and so the drug and formulation itself may explain why a result is positive or negative. And then patients, of course, are all unique. Everyone has unique pharmacogenetics based on genetics, clinical status, drug-drug interactions or herb-drug interactions. And so this is also an important thing to consider when evaluating a result that was maybe unexpectedly negative or unexpectedly positive. So the most likely approach in a pain management setting is going to include random urine drug testing. So a random specimen is collected, and ideally that screening process occurs close enough to the patient that the physician or caretaker can actually discuss those results with the patient to identify whether there's any concern regarding the results that are identified. When there is concern for one reason or another, that results are inconsistent with expectations, then confirmation testing may be indicated. So what do I mean by confirmation testing? Confirmation testing is generally a second technology that is very specific, very sensitive, designed for the drugs or the drug class of interest. Most of the confirmation methods are based on chromatographic separation and then mass spectrometric detection. At ARUP, we offer GCMS, LC-MSMS, and some LC time of flight mass spec as tools for very sensitive and specific confirmatory testing. But it's important to know that there's very little standardization across the industry in terms of those confirmation tests, so you need to know what you're getting ahead of time to make sure that it meets your needs. So to reiterate the main reasons to confirm results: first, when the screen results are inconsistent with expectations; second, when there are concerns about specificity of the screen that was used; and third, when quantitative results are necessary for appropriate interpretation of results. And so I'll give you a couple of examples at the end, near the end, about when quantitative results might be very useful. So this approach to testing is just illustrated graphically on this slide. So we start out with a screen, then evaluate the results. If the results are appropriate and the patient is very low risk for aberrant drug behavior, then there's really no further action required. If the results are not appropriate based on the clinical and behavioral scenario, then we need to consider carefully the drug that that patient is taking or drugs, the patient themselves, what their risk behavior profile might look like, the specimen, is it of high quality, is it timed appropriately, and the test performance characteristics relative to what was actually done. Confirmation testing may be indicated, and then we do it again, evaluate those results. If they're appropriate, then we're done. If they're not appropriate, then the investigation would continue. So investigation of results to make the correct interpretation is very, very important. The consequences of an incorrect interpretation are significant to everybody involved. So we want to do the best job we possibly can. And so I'm going to spend the rest of the time talking about some of the considerations surrounding interpretation of results, and in particular evaluating negative and positive results. So, starting with negative results, the most obvious interpretation when a result is negative is that drug was not administered. The patient did not take the drug. But before accusing the patient of not taking a drug they should have taken, we need to consider some other things. So if the result is unexpected, maybe it's because the patient's taking the drug incorrectly, such as less than prescribed or less frequently than prescribed. Maybe the drug delivery of the particular formulation that's been prescribed is variable and may help explain why that result was negative. The drug may not have been absorbed. Many drugs used in pain management have very wide ranges of bioavailability, so this is a plausible explanation. The patient, him or herself, may have accelerated metabolism or elimination. The urine specimen may be too dilute, or it may be adulterated. The test that was performed may not have been designed to detect that drug of interest, and so this gets back to that specificity issue and is a reason why sometimes we want to confirm negative results to make sure that that drug actually was not there. And then although we never want to admit it, there is a possibility that there was a mix-up somewhere. So before accusing a patient of not taking an expected drug, it's very important to consider all the possible explanations and confront that patient with a discussion. When we have a positive result, the most obvious explanation, of course, is that the drug was taken, that the drug was administered as prescribed. If that positive result is unexpected, then we have to investigate if there was a past prescription for that drug, if the patient's getting prescriptions from another clinic, if the correct prescription, of course this is unlikely, but it is possible that the prescription was not filled correctly. Now what clinicians are very concerned about is whether patients are taking non-prescribed drugs. And so this would suggest that there's a possibility. We have to explain or explore all possible explanations. Some things we might not think about is that the drug that was detected in the confirmatory test may be a metabolite of a prescribed drug. And I'll give you an example of that. It's also possible that it may be a process impurity of the pharmaceutical product itself. And I'll give you an example of that as well. It's also possible that the patient is trying to mimic compliance and so is adding drug directly to the urine. Now this is something you may not pick up easily on a screen, but with looking at quantitative results, it becomes quite evident. Another possible reason for an unexpected positive is that the test performed has poor specificity, so is a false positive. So again, investigate, interrogate the testing process. And of course, look at the clinic and lab to make sure every duck is in a row. So I wanted to provide a couple of examples for these most unexpected things, starting with drug being added to the urine directly after urine collection. And so this is just some data that we have collected from our urine buprenorphine confirmation test. And so what I've got here are buprenorphine concentrations in the first column, then norbuprenorphine, which is a common metabolite. Now in a patient that has taken buprenorphine, usually you see almost no buprenorphine and mostly norbuprenorphine. So this is a very unusual pattern and prompted us to investigate a little bit further. One of the things we did was recognizing that buprenorphine is often co-packaged with naloxone in Suboxone preparations, we thought, well, let's look at naloxone as well. So when we look at the concentrations of buprenorphine and naloxone, and these are free drug concentrations, which really should not be in urine, and then take the ratio, that ratio closely approximates what we would expect to see in the pharmaceutical preparation, thereby providing strong evidence that these patients simply added some crushed tablet to their urine, or perhaps used a sublingual film to dissolve some buprenorphine and naloxone in their saliva and then spit into the urine, something like that, to actually add the drug directly to the urine to mimic compliance. So this is a problem, and this would be one reason to do quantitative testing rather than just a screen. This is another example where quantitative results are useful. So if we find both morphine and hydromorphone in a urine specimen, we could conclude that a patient took both those drugs independently. But if we look at the concentrations, we'd see that the hydromorphone in this case is very small proportionally to the morphine concentration. And it's actually an expected pattern because hydromorphone is a minor metabolite of morphine. And that's illustrated in this schematic. So you can see that morphine, by way of the yellow arrow, goes to hydromorphone in a small quantity. And so we would expect to see some proportion of hydromorphone in a urine collected from someone that has been taking morphine. The actual ratios is somewhat variable from patient to patient and based on the timing of urine collection relative to the administration of the morphine, whether they take it chronically or just one time. So it's difficult to have a hard and fast ratio to say this means the patient did not take hydromorphone in addition to morphine, but if the hydromorphone concentration was significantly higher than the morphine, then I suppose you might conclude that. Okay, so now we'll complicate the picture and add codeine in the mix. Now codeine is not a metabolite of morphine or hydromorphone. In fact, codeine is metabolized to morphine, so if codeine was the source of the morphine, we'd expect codeine to be in much higher concentrations than the morphine. Generally, people only metabolize about 10% of codeine to morphine. So this pattern doesn't fit well, but it might raise some concerns with physicians about whether patients were taking hydromorphone, codeine, and morphine independently, or whether these results can be explained another way. So codeine, it turns out, can be an impurity in morphine preparation, so in the drug itself. And according to Medical Review Officer alert, up to 0.5% of morphine may actually be codeine. And these are some examples of other process impurities that exist. So we do in the lab see these patterns quite frequently, and it's important that clinicians not misinterpret and assume that a patient has taken more than one drug, when in fact this would be a normal phenomenon based on the sensitivity and wide analytical measurement range of these particular tests today. So in summary, unexpected results should stimulate an investigation of the test, the drug, the patient, and the sample. So the last thing I wanted to do was just talk a little bit about drug adherence versus dose and dosing adherence. So clearly we can determine whether a patient has recently taken a drug, but can we determine how much of that drug they took or at what frequency? Did they take it 3 times a day as they should? Or are they taking it all at once just before they come into the office? And unfortunately, I do not feel that urine drug testing can reliably evaluate dosing. And these are some of the reasons why. First of all, dose delivery does vary with formulations, and there's a number of literature sources I can reference you to that would provide examples where dose delivery will vary with formulation. We know that pharmacogenetics varies by patients, so to back calculate from concentrations to original dose is very difficult. We're also challenged by the fact that most specimen collection for this type of testing is not timed, and if we were going to time it, we would have to find out when the prior void was voided and when the collected void was voided. So it's something that people don't normally keep a log of, and we don't have access to. We know that urine varies based on hydration status, and so getting a creatinine concentration may help us normalize the result for an individual patient to provide some degree of serial monitoring, but it's very difficult to then, like I say, back calculate to an actual dose or dosing frequency. We also know the amount of a particular dose that's eliminated in the urine varies, and that most testing is actually based on measuring drug breakdown products, not the parent drug itself. So that complicates the entire picture. Finally, we know that routine chronic drug administration will affect the amount of drug and drug metabolites that appear in the urine and for how long they appear. And as I mentioned, laboratory tests vary, so it's very important to talk to the lab that's doing the testing to find out what the strengths and limitations of that testing protocol are. So in conclusion, thank you for being so attentive and patient. I do believe urine drug testing is very useful in the pain management setting to evaluate drug use. Testing technologies and frequency of testing should be aligned with clinical needs and expectations. Results have to be interpreted carefully in the context of the test, the drug, the patient, and the individual sample. This is particularly of concern when we have unexpected positive or negatives, and confirmation testing may help resolve that concern. Alternate specimens may also help resolve concerns. And so don't exclude the possibility of doing something like blood testing. And then finally, that dose and dosing of a drug really can't be reliably determined by urine drug testing alone. So thank you very much. And